everyone and welcome to my video lecture. So today we are going to start with a new chapter which is called as evolution. Okay, and in the state board textbook it is chapter number 5. Now for this year because of the COVID-19 crisis, most of the units of this chapter have been deleted, so they are going to be non-evaluative for 12th standard. Okay, so there are 13 units in this chapter, 5.1 to 5.13. Out of them, there are only 2 units which are there for your 12th standard exam, and they are unit 5.6 and 5.13. Okay, so all the other units apart from 5.6 and 5.13 they have been deleted. Please remember. So we have to study only unit 5.6, and that is modern synthetic theory of evolution. This is unit 5.6, and the last unit of the chapter 5.13, which is human evolution. So you can say that almost 95% of the chapter or 90% of the chapter has been deleted for you for 12th standard. Okay, so we will be starting with this first, and then we will be starting with human evolution. Human evolution is a very small part. Okay, so let us start with unit 5.6, which is called as modern synthetic theory of evolution. So let me just write it down over here. Is this clear? Alright, so now I will start with that. Now, what is modern synthetic theory of evolution? Now, before this theory, there have been many other theories also, since that part has been deleted. For example, there is a theory called as chemical theory of evolution. Okay, chemical theory of evolution was the first proper theory which was given. It was given by 2 people called Biren and Haldane. Okay, then there was Darwin's theory. Darwin's theory of natural selection was there. Alright, origin of species, that was the name of the theory. Then there was mutational theory by Hugo de Vries. So numerous theories have been formulated before this theory, but what happened was each of these theories had some drawback. Okay, कुछ ना कुछ तो वो theories के अंदर drawback था. Now this modern synthetic theory of evolution, it is the latest theory, and basically it takes the positive points or the plus points of all the other theories without taking into consideration their drawbacks. So वो drawbacks को हटा के जितने भी plus points है from the other theories, they have been taken in this modern synthetic theory of evolution. Also, there is proper evidence or there is proper proof for this theory. For example, there are many evidences. There is anatomical evidence. What is anatomical evidence? Anatomy is basically the structure of the body, the structure of the organism. Okay, so that is an anatomical evidence. There is paleontological evidence. What is paleontological evidence? Evidence based from fossils. So if we find the fossil of an organism, then that is the proof that that organism was existent at some point, right? Then there are biochemical evidences, there are molecular evidences, there are many proofs about this theory. Is this clear? So we are going to start this theory. This is the biggest theory in this chapter, and you only have to study this for your 12th standard exams. Okay, now who proposed this theory? So each theory is basically formulated by people. Okay, for example, as I told you, chemical theory was given by Biren and Haldane. Okay, mutational theory was given by Hugo de Vries. Okay, similarly Darwin gave his theory of origin of species. So this theory was not given by some single person, it was given by many people combined together. Most of the researchers at that time contributed in this theory. So I will write down some of the names who were responsible for this theory. Okay, so Simpson, okay, then Huxley, okay, then somebody called Wright, alright, then Mayer, okay, then Morgan, then Dobzhansky. So many examples are there. Okay, you just remember 2 to 3 words, 2 to 3 names, and that is enough. So all these people have together formulated this theory called as modern synthetic theory of evolution. So right now we follow this theory of evolution. Is this okay? Okay. Now this theory of evolution basically is divided into three main parts. Okay, we are going to study that. The first part of this theory is called as genetic variation. The second part is called as natural selection, and third is called as isolation. So there are three parts of this theory. Before we start with those three parts, there are three words which you should be knowing. There are three terminologies which you should be knowing. Okay, because if you don't understand this three terms, then it will be difficult for you to grasp the theory. आपको समझेगा नहीं वो theory. Okay, and what are these terms? So let me write down the first term which is important for understanding this theory. That term is called as a Mendelian population. What is the meaning of Mendelian population? Please pay attention. Okay. First of all, what is a population? So when we are speaking of evolution, a population is a group of organisms belonging to the same species. Please remember. Okay. For example, if this is an area, alright, and these are the organisms that I am drawing, then all these organisms, if they belong to the same species, then this entire group is called as a population. So population is basically all the organisms belonging to the same species. This is in relation to evolution, please remember. Okay, now what is Mendelian population? Mendelian population, so just imagine that these organisms belong to the same species, so they can interbreed. Just imagine in this population, if all the organisms are tigers, okay, then they will interbreed with each other. So a group of population or a population which is having interbreeding organisms, interbreeding means they can breed or they can mate with each other. Such a group or such a population is called as a Mendelian population. So basically, when this term comes in front of you, Mendelian population, you have to understand that that means that the group of organisms that is in front of you, it may be plants, it may be any organism, all those organisms can interbreed with each other. Suppose if I say that all these organisms cannot interbreed with each other, that means it is not an Mendelian population, it is just a population. Because you can have all the organisms of the same species, but there can be no breeding between them, then that will not be a Mendelian population. So Mendelian population means group of organisms which form a population and which can interbreed with each other, or they can mate with each other, they can reproduce with each other. Is this clear? So this is the first thing that you should know. The second term which is important for modern synthetic theory of evolution is the term which is called as gene pool. Okay, this is again a very important terminology. Everybody knows what a gene is, so I hope everybody knows what a DNA is, right? So suppose if I draw this DNA, okay, and this is 5 prime to 3 prime, correct, and this is 3 prime to 5 prime, okay, and I am writing down random nucleotides A, T, G, C, A, G, C. Over here the sequence will be T, A, C, G, triple T, C, G, correct? Now what is a gene? A gene is basically a part of DNA. So I can say that this part is gene number 1, this part is gene number 2, this part is gene number 3. So basically all the genes together make up the DNA. Okay. Now what is a gene pool? So everybody has understood what genes are. Genes are small fragments or parts of a DNA. Now DNA is condensed to form chromatin, and chromatin is further condensed to form chromosome. Correct? So genes are present on the chromosome. Now what is gene pool? So now you know what a Mendelian population is. Correct? So again I will draw the same thing. Suppose if these dots which I am drawing, suppose if they are all organisms of a Mendelian population, means all these organisms can interbreed. Now gene pool is equal to the total number of genes present in this population. So if I want to find the total number of genes present in this population, how will I find that out? Very simple. I have to total the genes present in each organism. So if this organism has some genes, then this has genes. So I have to calculate the total number of genes present in all the organism, and that total is called as the gene pool. Is this clear? Okay. For example, just imagine that you are sitting in a classroom. Okay. There are 29 total. There are 30 total students, including you, and there is one teacher. So total, how many people are there in the classroom? 31. If I say that calculate the gene pool of the classroom, then what will you do? Very, very simple. I will have certain number of genes, you will have. So each organism or each person will have certain number of genes. The total of all those genes together is called as gene pool. Is this clear? So gene pool is the sum total of all the genes present in a Mendelian population. See, it is very important to understand these words because in NEET, many questions are asked on these terminologies. If you by heart or if you mug up the concept, then you will not be able to explain it. So please understand the concept. What is the meaning of Mendelian population? What is the meaning of a gene pool? Okay, if you have understood gene pool, then the next concept is called as a gene frequency. This is the last terminology which you have to understand before we start with the theory. We have still not started with this theory. For this theory, you have to understand these three concepts, then we will be starting with the theory of modern synthetic theory of evolution. Okay, now what is gene frequency? Now, many people get confused over here. Okay, so I am going to give you an example which will make you understand this theory better. Alright. Now, again I am going to draw a population. Okay. So, this is a population. Now, this is not a Mendelian population. Okay. This is just a random population. So, just imagine I am using. So, these are the organisms that I am drawing in blue color. Okay, there are how many? 1, 2, 3, 4, 5. This is organism number 1, this is number 2, this is number 3, this is number 4, this is number 5. Okay, then I am drawing another organism with red color. Okay, so this is number 6, number 7, number 8, 9, 10, 11, 12. So just imagine all these are cows, all these are wolves, and now I'm going to draw a third type of organism which is going to be green color. So 13, 14, 15, 16. Now if I ask you that all the organisms belonging to this blue colored category, what is the frequency of these organisms? Now, what do you mean by frequency? Frequency means the proportion of these organisms in this entire population. Okay, so right now, if you ask me how many total organisms are present, then total organisms which are present are 16. What is the proportion of these organisms in this population, or what is the frequency of these organisms? Then the frequency is 5 upon 16. Very simple. So, are you understanding what frequency is? Frequency is basically the proportion of that organism or that substance with respect to the total population. Okay guys, so I hope you have understood what the concept of frequency is. So now what is gene frequency? So in a similar way, gene frequency is basically the frequency of a allele at a particular locus. Locus is what? Locus is the position in the chromosome. So gene frequency is the proportion of an allele at a particular locus when it is compared to other alleles. Okay, so suppose if this is a chromosome, alright, and now suppose if I want to find out the gene frequency of this gene, okay, there is one gene. So what I do is I compare this gene with all the other genes present in this area, and that proportion is called as the gene frequency of that gene. Is that clear? Okay, so that is called as gene frequency concept. Please remember, normally the frequency of the gene remains more or less same. Okay, the gene frequency doesn't change much. Slight fluctuations are possible, but a very large change doesn't take place in a population. What is a population? A population is basically a group of organisms belonging to the same species. What is a Mendelian population? Mendelian population is a group of organisms belonging to the same species and which can interbreed. Okay, what is gene pool? The sum total of all the genes present in the Mendelian population is called as the gene pool. And what is gene frequency? The proportion of one allele in a chromosome at the same locus, when it is compared to the other genes in that locus, it is called as gene frequency. Is that clear? Okay. So now let us start with the modern synthetic theory. Okay. Now under modern synthetic theory, there are three important parts which you have to learn. Okay. The first part that we will be covering right now is called as genetic variation. Okay. This is what we are going to cover right now. Then the second part of this theory, alright, it is called as natural selection. And the third part of this modern synthetic theory is called as isolation. Okay, it is also the last part. Is this clear? So genetic variation, natural selection and isolation are the three parts of the modern synthetic theory. This is one of the biggest theories and this is the only theory which is there for your 12th standard for this year. So please pay attention now. Alright, now what is the meaning of genetic variation? Genetic variation simply means that there is change in the gene frequency. Okay. What is change in gene frequency? That means the proportion of that particular gene when it is compared to other genes that has changed. That proportion has changed. So genetic variation, we can also call it as a change in gene frequency. Is this clear? Okay. Now under genetic variation there are further types. So what I am going to do is I am going to rub all of this and we are going to start with genetic variation right now. Okay. So please pay attention. Very important part. Natural selection and isolation are very easy to understand, but many people get confused in genetic variation. So now we are starting with the first part under modern synthetic theory, and that is genetic variation. Is this clear? Now this genetic variation is further divided into many parts. So there are causes of genetic variation. Okay, again I am repeating what is genetic variation. Change in gene frequency is called as genetic variation. In simple words, the gene pool is getting changed because of the change in genes. That is called as genetic variation. Now, what is the first reason for genetic variation? The first reason that we are going to study is called as gene mutation. Now, all these reasons are very important. Okay. What is the meaning of gene mutation? What is mutation? Change in the sequence of nucleotides in a DNA is called as mutation. For example, suppose I am just drawing a single stranded DNA. This is the strand, and suppose if I write down the sequence A, T, G, C, A. Now this is the original sequence. Suppose if this sequence changes to, now instead of A, suppose this nucleotide changes to G, then the sequence will become GT, GC, AA. So now you can see that this sequence and this sequence are different because this first nucleotide has changed. This is called as mutation. Change in nucleotide is called as mutation. Is this clear? Why is this change happening? It is just happening randomly. Okay, mutation just happens at random. It is by pure chance. So this is called as mutation. Okay, now what is happening because of mutation? Just imagine this is gene number 1. This is your gene number 2. So your first gene is what? It is ATG. However, after mutation, your first gene has become GTG. So your first gene has completely changed. Your second gene still remains the same. It is CAA. So now you understand. The gene has completely changed because of a change in single nucleotide. This is called as mutation. Okay. This type of mutation is called as point mutation. Point mutation means there is change in only a single nucleotide. Apart from these two nucleotides, no other sequence has changed. Suppose if I change this T and I write it down over here as C. So now there are two nucleotides which have changed. This is not called as point mutation. Please remember, point mutation means only a single nucleotide has changed. So this will remain the same. Now because of gene mutation, what is going to happen is, see, genes are present on DNA. DNA gets condensed into chromatin. Chromatin gets condensed into chromosome. So basically what is happening is your chromosome structure is changing. So because of gene mutation, can I write that there is chromosome mutation? Yes, I can write down, because DNA and chromosome are the same things. So chromosome mutation is also taking place. And because of everything, what is happening is the gene frequency is changing. So that is the first part under genetic variation. The first factor causing genetic variation is gene mutation. Okay, so what is important to remember in this is, first of all, you should know what is gene mutation. Okay, change in nucleotide sequence of a DNA molecule is called as mutation. Then you have to remember what is point mutation. Change in a single nucleotide is called as point mutation, right? If I change two nucleotides, then it is not called as point mutation. Is this clear? Then what you need to remember is, chromosome is basically DNA which is condensed. So if there is mutation occurring in the DNA, obviously there is mutation occurring in the chromosome. So what is happening? The genes are changing. Just imagine this is the original population. Okay, this is the DNA of organisms in the original population. I will write it as population 1. After the mutation, this gene has changed into this gene because of this one nucleotide. So now the gene frequency of that same population has changed, because ये वाला जो gene था वो तो अब नहीं रहा, दूसरे में बदल गया. So now this population 1 will become slightly changed because of this change in nucleotide. Is this clear? So that is why gene frequency is also changing. So I hope this first factor which is causing genetic variation is clear. It is also called as gene mutation. Now, the next factor causing genetic variation is called as genetic recombination. Now, recombination, what is the meaning of that? Just think till I write this down. What is the meaning of genetic recombination? Have you learnt about crossing over? What is crossing over? So crossing over happens during meiosis, right? During packaging phase, if you remember. So what happens in crossing over? So if you want, I will just draw. These are the two homologous chromosomes, correct? They have undergone synapsis. Okay, now this is called as a tetrad or a bivalent, if you remember. Okay, and these are the two non-sister chromatids. Now what happens, if you remember prophase 1 of meiosis 1, the stage where these two homologous chromosomes attach to each other, that stage is called zygote. And then what happens is, there are genes present over here, alright. So I am drawing these genes in blue color. And there are genes present over here. I am drawing those genes in green color. I hope you are able to differentiate between these two colors on your screen. Now what happens in crossing over? So crossing over occurs in packaging stage. These genes will move to this side, these genes will move to this side. Are you getting me? So what happens is, after crossing over, suppose if I draw these chromosomes, then I will have the blue genes which will be present over here because they have crossed over to the opposite side, and the green genes will be over here because they have crossed on to the opposite side. So what is happening in crossing over? Just imagine the gene frequency of both the chromosomes is changing because of crossing over. Are you getting me? This crossing over is also called recombination. Why is it called recombination? Because पहले के जो combinations थे वैसे नहीं रहे. अभी यहाँ पे जाके ये पार्ट इधर रिकम्बाइन हुआ, ये पार्ट इधर रिकम्बाइन हुआ है. So that is a mixing which is occurring. That is called genetic recombination, or it is called crossing over. So obviously the gene frequencies will change. Are you getting me? Because what is going to happen later on, prophase 1 के बाद there will be metaphase 1. Anaphase 1 में both the chromosomes will separate. Are you getting me? And each chromosome will go to a different gamete. So now each gamete is having a separate, having this chromosome but now the original genes are not present. This recombination of genes are present, and that is very important for evolution, or it is very important for variation. And this is the other factor which is causing change in gene frequency, because your original gene order is changing. Because now if you take any gene among these genes, तो उनका जो ओरिजिनल लोकस था अभी वो चेंज हो गया, तो ऑब्वियसली जीन फ्रीक्वेंसी विल चेंज. So second reason for change in gene frequency, or second reason for genetic variation, is called genetic recombination. So you have to remember, under genetic recombination you have to basically explain what is crossing over. Okay, and crossing over is very important for variation. Variation is very important for evolution. Okay, are all the human beings alike? No. Why? Because of variation. Are you exactly a same copy of either of your parents? हम लोग exactly mother या father की तरह नहीं दिखते. Why? Because of variation. अगर variation नहीं होता था, crossing over नहीं होता था, तो आप exactly दूसरे कोई तो इंसान की तरह copy रहते थे. Are you getting my point? So the second point, is this clear? Okay, genetic recombination is another reason for genetic variation or change in gene frequency. So I am going to write down over here, this is responsible for variation and it is also important for evolution. See, we are basically studying the first part under modern synthetic theory, and that first part is genetic variation. Now under genetic variations also there are factors. We have studied two factors. Now let us come to the third factor. The third factor which is causing genetic variation is called as gene flow. What is gene flow? See, very simple. Genes flow from one population to other. Dekho, I am just drawing. This is one population over here. Okay, and I am drawing the organisms. So this is one Mendelian population. All the organisms are in blue. Now what I am drawing is, this is another Mendelian population, all the organisms are in red. Now just imagine, some organisms from this population, they migrate over here. So I will rub these three organisms from here and I will add them over here. So now what is happening is, these organisms have migrated from their original population to this population. So now what happens in this population, the genes have changed. Now the gene pool has changed because three organisms have been added. What is gene pool? Gene pool is sum total of all the genes of organism. So if three organisms have been added, the gene pool will change. If the gene pool changes, the gene frequency will also change. Common sense. So this is called as gene flow. In a similar way, organisms from here, they may migrate. So suppose if I say three organisms leave this population, they go over here, then the gene pool of this population will also change, gene frequency will change. Please remember, if the gene pool of the population changes, it automatically changes the gene frequency. Because the genes in that particular gene pool, that gene frequency is counted with respect to all the other genes, but here genes are only changing. Obviously gene frequency will change. Is this clear? So gene flow means what? The gene migrates from one population to other. It is normally due to migration of animals. Okay, I will give you an example. During winter in Russia, okay, so what happens is, there are Siberian cranes. So these Siberian cranes, when it is winter over there, they migrate to Bharatpur, which is in Rajasthan. Then again after some time they fly back. So that is migration. So if you take the gene frequency of Siberia during winter, it has changed because the cranes have migrated. Are you getting me? Other example of gene flow in case of plants is, you must know that some plants, they release spores. So that spore, if it is carried by wind to some other population, again the gene frequency will change. Are you getting this point? So is this gene flow clear? So gene flow is another reason why there is change in gene frequency or there is genetic variation. Best example is migration of organisms. Clear? Next, coming to the fourth reason causing genetic variation. Okay, so what I will have to do is, I will have to rub this off because I don't have space. So the fourth factor causing genetic variation is called as genetic drift. Alright, so now This is the fourth reason which is causing genetic variation. Which are the reasons we have already studied? कौन से reason genetic variation कर रहे हैं? पहला है gene mutation. Simple. Second is genetic recombination, right? Crossing over. Third reason we studied was genetic gene flow. That is due to migration of organisms. Simple. One organism or many organisms leave that population, go to other population. Now gene pool of both the population has changed, so the gene frequency of both the population has changed. Simple. Now we come to genetic drift. Now genetic drift. Now many people get confused between gene flow and genetic drift. Now please pay attention. Genetic drift is also a change in gene frequency. But this is happening randomly. Randomly means there is no specific reason for that. Okay. Now if I talk about gene flow, in gene flow also genes are leaving one population, they are entering other population. So the gene pool is changing, gene frequency is changing. But gene flow is occurring due to a reason. For example, animals will always continuously migrate. So that is why gene flow is occurring. But now just assume that this is a population, these are the organisms in that population, so this is the Mendelian population, they can interbreed. Now just imagine in this area, suppose there is a natural calamity, okay, suppose a cyclone occurs, and because of cyclone, just imagine that half of the population dies, okay, cyclone has occurred, and because of the cyclone, half of the organisms in this Mendelian population have died. Now what will happen to the gene pool? Obviously the gene pool will change, because the organisms which are dead, they don't have, those genes are not counted. So now only these living organisms, their genes will be counted, the gene pool will change. Gene pool will change, obviously gene frequency will change. But now has this occurred because of some specific reason? No. It has occurred because of cyclone. Does cyclone always come periodically? No, it has just come by chance. Are you getting me? So this is also change in gene frequency, but this change in gene frequency is purely by luck or purely by chance, or it is happening randomly. Random मतलब it is happening for no reason. कोई भी चीज repeatedly होती है उसको random नहीं होता, उसको non random होता है. Random मतलब अचानक से हो गया. Other example is there is a forest fire. Some organisms have died. The gene pool has changed, gene frequency has changed. Does it always happen? No. So that is random. So that is genetic drift. So gene flow में भी genes alter होते हैं, gene pool alter होता है, gene frequency alter होता है. But difference between gene flow and gene drift is in flow. In the gene flow, the organisms normally migrate, and because of that, the gene leaves one population, enters other population. In genetic drift, the genes are not leaving one population and entering other population. Only the genetic pool of that population is changing, and because of that, the gene frequency is changing. Another example of genetic drift is death. If the organism dies, so what happens is the gene pool changes, the gene frequency changes. Now, can you tell me one thing? Genetic drift will basically affect which population? Larger population or smaller populations? See, if the population is very, very large, if there are many organisms, just imagine thousands and crores of organisms, okay? Many organisms. So this is a huge Mendelian population. And suppose in this population one organism dies. तो एक organism के मरने से gene pool ज़्यादा change नहीं होगा, एक ही organism का है ना? Are you getting me? But if I draw another population which is having only 3 organisms, and here if one organism dies because of death, तो यहाँ पे gene pool बहुत change हो जाएगा, because already the organisms are less. So genetic drift normally affects which populations? It affects Mendelian populations which are smaller. It doesn't affect the larger populations to that extent. So please remember this, genetic drift is caused randomly. Randomly means events which are not occurring continuously or cyclically or for a good reason. They are just happening suddenly, and because of that the gene pool and frequency changes. Examples of calamities are natural calamities. So natural calamities, you can give examples such as cyclone or earthquake or you can also write tsunami, or you can write forest fire, or you can even write death. So here you have to remember the genes are not flowing from one population to other. No, in gene flow the genes are flowing from one population to other. Here there is no flow. Here also the gene frequency is changing. In gene flow also the gene frequency is changing. In both the cases the gene pool is also changing, but this is occurring randomly. Please remember. Okay, and this is going to affect smaller populations more. If the population is very large, it doesn't affect the population much. Just imagine 1 crore is the population of a Mendelian population. If 1% dies in that, gene pool को ज़्यादा नहीं फर्क पड़ने वाला है, hardly insignificant change है. Is this clear? So this is called as genetic drift. Okay, and genetic drift is also called as Sewall Wright effect. It is not given in your book, or the part when it is given that has been deleted, but this is important for me. So genetic drift is also called as Sewall Wright effect. Now Sewall Wright effect says that genetic drift normally affects a smaller population. Affects means it affects the gene pool and gene frequency to a large extent. Whereas if there is a very large population, then genetic drift doesn't make much of a change to the gene pool and gene frequency. Is this clear? Gene flow and genetic drift में difference clear है? ये भी genetic variation कर रहा है, gene flow भी कर रहा है. Last factor causing genetic variation is called as chromosomal aberration. Alright, so let me write it down over here. The last factor causing genetic variation is called as chromosomal aberration. Now what is the meaning of aberration? Aberration. Aberration means damage. Okay, please remember, aberration means damage. So chromosomal aberration means there is a structural damage to the chromosome. So the shape of the chromosome might change, the structure of the chromosome might change, the nucleotide sequence obviously it will change. So that is called as chromosomal aberration, a damage done to the chromosome in either structure, shape or nucleotide sequence is called as chromosomal aberration. Now what are the different types of aberrations? So you have got 4 examples under chromosomal aberration. Okay, let us study them. The first type of, obviously chromosomal aberration will also cause mutation because it is going to cause a change in your nucleotide sequence. Okay, so now first kind of chromosomal aberration is called as addition. Now what is addition? Very simple. To a nucleotide sequence an additional nucleotide will be added. I will just write down the example. Okay, so the original sequence I am writing 5' to 3' A G C T A A. Now what happens after chromosomal aberration in addition is, in this nucleotide sequence anywhere an extra nucleotide will be added. For example, if I make this sequence into A G C T, now between this T and A I am adding another A, so it will become A. Is this clear? Sorry, I wrote an extra A over here. So this will be CT A. Now this A is an extra A. It has been added. So this is changing the shape of the chromosome because the nucleotide sequence has increased. So this will cause a change in DNA, it will cause a change in chromatin, it will cause a change in chromosome. So this is called chromosomal aberration. It is also mutation, same thing. If I ask you which type of mutation, this is point mutation. Why? Single nucleotide has changed. Are you getting the logic? Okay. Next, we come to second type of chromosomal aberration, which is called as deletion. Now, in deletion, you will delete nucleotide sequences. See, in addition, you can even add more nucleotides. For example, if I say that between this T and this A, G and C have been added, then it will become TGCA. So addition can be any number of nucleotides. But if only a single one is added, it is point mutation. If more are added, then it is not point. So addition में extra nucleotide sequence में add हो जाता है. Deletion में Similarly, you will delete. So again, same sequence. Let me write down A, G, C, T, A, 5 prime, 3 prime. After deletion, suppose if I delete all these three nucleotides, what will the sequence become? The sequence will then become A, G. These three I have deleted, and the last one is A. So this is 5 prime to 3 prime. All these three have been deleted. Simple deletion. Getting my point? You can delete one nucleotide or more than that. Again, the shape of the DNA will change. Finally, the shape of chromosome will change. The third type of chromosomal aberration is called as inversion. So I will just rub this off because I want more space. Okay. So addition we did. Second we did deletion. The third that we are going to do is called as inversion. Inversion means what? Just pay attention now. Again, I am drawing a sequence 5 prime to 3 prime, A, G, C, T, A. Okay. Now what I am doing is this sequence I am taking and I am rotating it by 180 degrees. So what will happen is this 5 prime, this 3 prime, A, G will remain the same, but now this will rotate by 180 degrees, A, T, C, A. So what happens is the nucleotide sequence reverses itself. Only this part will change its polarity, it will rotate by 180 degrees. This is called as inversion. Ulta ho jayega. Getting this? And the last type of chromosomal aberration is called as translocation. What is the meaning of translocation? See, translocation means a nucleotide sequence from one chromosome will break and it will go and attach to some other chromosome. For example, just imagine this is one chromosome, this is another chromosome. Okay, suppose if there is this sequence ATG over here and this ATG breaks. So what will happen is this part of the chromosome is broken, this ATG might go and attach over here. Okay, so this is called as translocation. So this chromosome will lose that sequence, this chromosome will gain that sequence. This is called translocation. So these are the 4 types of chromosomal aberration, and with that we have finished with the first part of modern synthetic theory of evolution, that is called genetic variation. Okay, now the next 2 points are very simple. One is natural selection, other is isolation. They are very easy, but many people have problem in this part because many words sound very similar. So for you to understand genetic variation, it is very important that you understand what is Mendelian population. Mendelian population, please remember, all the organisms in that population belong to the same species and they can interbreed, they can mate, they can reproduce. Second is gene pool. Gene pool is basically in the population, you take all the genes of all the organisms, you total them, that is called as the gene pool. And gene frequency, now gene pool you have, correct? You pick any gene from that pool, you take the position, you point the locus of that gene, and then in that locus you compare that gene which you have picked with all the other genes. That is called as its gene frequency. Is this clear? Now the first point under modern synthetic theory is genetic variation. Genetic variation means change in gene frequency. Which factors cause change in gene frequency? First is gene mutation. Gene mutation may you have to remember point mutation. Second factor causing genetic variation is genetic recombination, meiosis, crossing over. Third factor and fourth factor sound similar but they are different. Gene flow, genetic drift. What is the difference? Gene flow mein genes will migrate from one population to other because of migration of organisms. It doesn't occur randomly, it occurs continuously for some particular reason. But genetic drift occurs randomly, suddenly, no particular reason. Example, natural calamities, death. Genetic drift mein the gene is not leaving one population and going to other population. It will just leave that population, that is it. Are you getting me? And genetic drift normally affects smaller populations more. Affects means it affects the gene frequency of smaller populations more and it affects the gene frequency of larger populations less. Okay, and last point which affects genetic variation, or the last factor which affects genetic variation, is called as chromosomal aberration. We have studied all the 4 types, very simple: addition, deletion, inversion and translocation. Basically your nucleotide sequence is changing, so all this can also be called as mutation. Are you getting me? So we have finished with the first part of modern synthetic theory. Now I will make a separate lecture on natural selection and isolation. Okay, we will complete it in the next lecture, and I think after that will be the last lecture for this topic, where I will teach you human evolution. Okay, so if you have any doubts in this part, please let me know. Okay, please read this part from the textbook. It is very important for your 12th standard exams also. And thanks for watching the lecture.